Good morning. I'd like to thank Brent and Philippe for inviting me to participate in this wonderful setting and a very exciting topic for this symposium. This morning I'd like to begin to consider with you the importance of initial bacterial colonization in the development of mucosal host defense. To do so, I'd like to briefly consider with you first the programming of the gut, what is known about initial normal bacterial colonization and its effect on host defense from a developmental perspective. I'd then like to consider some conditions that lead to inadequate colonization and the clinical consequences of this. And then finally suggest to you that a potential solution for inadequate colonization is to use probiotics as a surrogate colonizer before I summarize and conclude. So let's begin with programming. On the left-hand side of this slide is a cartoon depicting the small intestine of the human fetus in utero. And as you can see, the epithelium is very thin, it's immature, and it turns over very slowly, and there's a paucity of lymphoid elements. In contrast, on the right-hand side of this slide is the same section of small intestine of an infant in the extrauterine environment. Here you see a robust, mature, rapidly turning over epithelium, which expresses all the subclasses of epithelial cells, and there's an abundance of lymphoid elements. The principal difference between these two environments is the presence of colonizing bacteria, and I want to try to develop that with you if I can. Let's begin by briefly reviewing initial bacterial colonization. Full-term infant leaves a relatively germ-free environment, passes through the birth canal, where it picks up a healthy bolus of bacteria from mother's vaginal and colonic cavities, and this represents the first phase of initial colonization. This bolus is stimulated by the beginning of oral feedings, and then is further modified at the time when the infant is weaned to solid foods, so that by about a year to 18 months, the intestine is completely colonized with the signature microbiota that's unique to that individual and in general remains with that individual the rest of their life. What happens under complete colonization is there's an enormous number of microbes that exist in the intestine, mostly as anaerobes in the colon and distal small intestine. In fact, there are about 10-fold more microbes in the intestine than there are cells in the body. In addition to the anaerobes in the colon and distal small intestine, there are facultative anaerobes and aerobes that exist in the rest of the intestine. And the numbers of species, as we have become more acquainted with the microbiota, is ever increasing. It's now thought that there's probably more than 1,000 different species of bacteria that reside within the intestine. A number of host defenses are developed as a result of colonization, and I just wanted to briefly consider with you a somewhat new concept, colonization resistance, which is defined as by virtue of gut colonization, microbiota can be a protective force against the invasion of pathogens. So what happens is the intestine is colonized with large numbers of microorganisms that are interacting with other organisms and the epithelium itself. And as a result of that coat of commensal bacteria, the pathogens cannot attach and penetrate through the intestine. During the first several phases of colonization, the nature of the microbiota is influenced a great deal by the environment. Let me sort of illustrate that, and that in the context of how the infant is initially fed. If an infant is breastfed, during a period of breastfeeding, there's an abundance of health-promoting bacteria such as bifidobacteria and lactobacilli, in contrast to a non-breastfed infant that has a more mature microbiota. Now, why is that important? Well, there's a recent publication that suggested that the presence of bifidobacteria at 1 month of age correlates directly with the level of secretory IgA in the intestine, suggesting that its presence is a stimulus for production of that immune globulin. Also, in the same publication, there's evidence to suggest that the presence of Bacteroides fragilis at 1 month influences what has been seen, and I'll point this out in a little more detail in a moment, the excessive inflammation that occurs to stimulation as measured by IL-6. So there's an inverse relationship between this organism and the level of inflammation. And you're going to probably hear more about this later on in this symposium from Dennis Casper. What happens under normal colonization circumstances is that a balanced ecosystem exists with health-promoting bacteria containing potential pathogens, and there's no expression of disease. Now, what about colonization and host defense? We know that bacterial colonization influences a number of functions of the developing intestine. Probably the most important function of colonizing bacteria is its influence on the development of mucosal host defense. And this was very nicely depicted in a review, in Nature Review by David Artus, suggesting that the presence of colonizing bacteria can influence the expression of microvilli. The presence of tight junctions can cause the glycocalyx to form as an additional barrier to the penetration of pathogens, can result in production of antimicrobial peptides, can influence the expression of M cells, and I'll come back to that in just a moment, and also affect dendritic cells both within Peyer's patches and within the intestinal interstitium. What happens is that colonizing microorganisms interact with the underlying epithelium, as shown here in this electron micrograph, and underlying lymphoid elements. And this is done through conserved molecular patterns that are principally expressed on the surface of the colonizing bacteria, which interact with pattern recognition receptors. As we all know, the most well-studied family of pattern recognition receptors are the toll-like receptors, and they can exist on the luminal surface of the epithelium, on the basolateral surface, and within the cells itself. The most studied of the mechanisms is the interaction of lipopolysaccharide and gram-negative organisms with the TLR4 receptor, which affects adaptive proteins and kinases, leading to the translocation of NF-kappa B into the nucleus and the expression of an innate inflammatory response. Now, we know that these exist for pathogens, but over the last several years, there's strong evidence to suggest that commensal bacteria, in this case commensal LPS, can interact with the TLR4 receptor to produce cytoprotective effects. We also know that the commensal CpG can enter the dendritic cell and interact with the intercellular TLR9 receptor and preferentially produce T regulatory cells and TGF-beta. Let me just briefly illustrate for you the impact of colonization in a generalized immune response. And then I'll come back to this in a moment with the innate immune response. This cartoon comes from a review article we wrote several years ago where we looked into the literature and attempted to associate in humans the development of various components of the mucosal immune system as a function of gestational age. And as you can see, virtually all of these functions are fully developed at birth. However, for them to be activated, they must first be interacted with colonizing bacteria, which affects cytokine production by lymphocytes within the interstitium and interepithelial, alters the composition of subclasses of T helper cells, stimulates the production of secretory IgA, and helps produce a specialized form of epithelium. Now, I was fortunate enough in the 90s to have a sabbatical, initially at the Pasteur and then at Curie with Daniel Levard. And an investigator working with me at that time was a young man named Eric Pringle, and he made what I think is a phenomenal observation. I mentioned the M cell. This is a specific form of epithelium which lacks developed microvilli and has a very little glycocalyx and facilitates the transport of antigens and microorganisms into lymphoid elements within the intestine. Now, what Eric showed was taking intestinal epithelial cells, exposing their luminal surface to microorganisms, commensal organisms, and their basolateral surface to lymphoid elements, and showing a transdifferentiation into the M cell itself. Example of how colonization influences the gut. Now let me just also illustrate a couple of examples of the influence of colonization on the innate immune response. What happens innately in the mature intestine is that pathogens interacting with the epithelium will activate the release of inflammatory cytokines and chemokines, which allow PMNs to come to the site and prevent the penetration of the bacteria. To prevent us all from having chronic inflammation, however, this is a self-limited process. And what has been shown is that the toll receptors interacting with molecular patterns, causing signaling to occur, resulting in an increase in inflammation, with sustained interaction can also stimulate negative regulators, which at various steps along the way can turn off the response so that it's a self-limited one. In addition, we know that mature enterocytes can discern between pathogens and commensal bacteria, causing inflammation only with the pathogen, and that commensal bacteria can secrete products which can influence inflammation, such as the ubiquitization of phosphorylated NF-kappa B. Let me just take a moment because I know Nadine Surf-Benison will probably be talking about this in more detail to illustrate the influence of colonization on adaptive immunity, using as examples, again, polymeric IgA secretion, a balanced T helper cell response, and the phenomenon of oral tolerance. At birth, all full-term normal infants are IgA deficient. It takes a finite period of time in the postpartum environment for protective levels to develop. I've already mentioned that bifidobacteria seems to influence this phenomenon, but it's interesting that the time span for the development of protective levels corresponds to the first and second phases of normal colonization. Another speaker later today, Andrew McPherson, published in Science in 2004 the mechanism by which IgA is produced. And what Andrew suggested is that dendritic cells that penetrate between epithelia or underlie the M cell can take up commensal bacteria, present it to B cells so that they become IgA-producing plasma cells to coat the intestinal surface. This was also shown to be the case for the distribution of T helper cells. In a very seminal observation made in Nature, it was shown that submucosal dendritic cells can extend their appendage between the epithelium and through expression of receptors sample commensal bacteria within the intestine. As a result of that interaction, they can release cytokines which create a microenvironment that produces a balanced T helper cell response so that all aspects of adaptive immunity exist. Delayed type hypersensitivity, clearance of pathogens that have been taken up by cells, antibody-producing humoral immunity, and very importantly, regulatory processes producing oral tolerance and anti-inflammation. I mentioned at the outset of this lecture that the intestine of the human infant in utero compared to the infant in the extrauterine environment is strikingly different. That same observation has been made when you look at the intestine of a germ-free animal and a conventionalized animal. So we can learn a lot experimentally about the influence of colonizing bacteria by looking at germ-free and conventionalized animals. Now, the seminal initial observation published by the Gordon Laboratory in Science showed that a germ-free animal, which was colonized with a single organism, Bacteroides theta iota micron, could stimulate a number of genes within the intestine in areas of development, transport, and immune protection to cause a development, in this case, of the immune system. The same phenomenon has been seen in the concept of oral tolerance. Oral tolerance is defined as the downregulation of systemic immunity after oral exposure to harmless bacteria and antigens. Interestingly, and this is how it's seen, again, dendritic cells sampling epithelium stimulates naive helper cells to preferentially produce T regulatory cells which release TGF-beta and in this case turn off the humoral immune response. Interestingly enough, a very interesting publication in Journal of Immunology in the late 90s suggested that oral tolerance could not be achieved in a germ-free animal. What they did is they took a specific pathogen-free colonized animal and a germ-free, exposed it orally repeatedly to ovalbumin, and then looked at the development of an immune response. As you can see with the pathogen-free colonized animal, there's a progressive decrease, suggesting production of oral tolerance. In contrast, with the germ-free animal, tolerance could never be produced. Now, more importantly, another observation made was that when you take a germ-free animal and you conventionalize them, in this case, during the newborn period or during adulthood, you find that tolerance is more readily achieved when conventionalization occurs early on, suggesting that early on colonization is important in this phenomenon. All right, having briefly reviewed with you what happens under normal circumstances, let me take a few minutes and talk to you about inadequate colonization. There are several circumstances in which inadequate colonization occurs. The rapid delivery of the premature, the bypassing of the maternal birth canal with cesarean section, or turning off colonizing bacteria by the use of prophylactic antibiotics. In each of these instances, there's a sparse, inadequate initial colonization, and despite the stimulus of oral feedings and weaning, it takes a finite period of time for colonization to be attained, roughly 4 to 6 years, and during that time, the child is more susceptible to pathogens and immune-mediated disease is exemplified by allergy. Colonization resistance in this circumstance is not very adequate because there's not enough microbes to cover the intestine so that pathogens can pass through and create infection both locally and systemically. A very interesting recent publication looking at the composition of the microbiota in infants who are delivered naturally and those who are delivered by cesarean section. As you can see, those who are delivered naturally, the microbiota are very similar to mother's microbiota in the intestine. In contrast, the cesarean section delivered infant has a microbiota pattern which is more like that seen in mother's skin, suggesting again the absence of the initial phase of colonization. As a result of this inadequate colonization, there's an imbalance in the ecosystem and it favors pathogens leading to expression of disease. And let me just illustrate that with 3 observations. The effect of antibiotic use on asthma, cesarean section in allergy-prone mothers and the development of atopic disease, and then in some detail, some studies we've done in the area of necrotizing enterocolitis with prematurity. A paper published in the pediatric literature suggests a direct correlation with the number of incidences of use of antibiotics in the first year of life and the nature of the antibiotics and the development of asthma as a young child. Mothers who do not have a history of allergy delivered vaginally, if we ascribe a odds ratio of 1, we see that a mother who delivers vaginally but has a history of allergy has increased to about 2.5. However, mothers with a history of allergy delivering by cesarean section have almost an 8-fold increase in the development of allergy. We believe necrotizing enterocolitis is an example of initial inadequate nutrition leading to an imbalanced microbiota in prematures, resulting in the fulminant gastroenteritis that exists in this setting. This has been shown recently by examining the microbiome of premature infants acting as controls that didn't develop necrotizing enterocolitis and those that did. And as you can see, when you look at large families of microorganisms, the phylum, the distribution is fairly regular in the control, but is skewed in one direction in the patient developing necrotizing enterocolitis. In addition, if we look at the diversity of organisms colonizing the intestine, those that didn't have a much more diverse than those that developed it. And of course, in this, it was shown that patients with NEC received antibiotics. Now, a more important study recently done in 21 centers, NICUs in the United States under the auspices of Child Health and Human Development showed that there is a direct correlation with the number of days that antibiotics are used and the incidence of necrotizing enterocolitis. I mentioned earlier that the mature enterocyte can distinguish between commensal and pathogenic organisms and only produces inflammation when a pathogen interacts with the epithelium. Unfortunately, this is not the case with the human fetal intestine. And we showed this when we compared adult enterocytes with our fetal cell line, showing, as expected, with a pathogen, a salmonella pathogen, an increased inflammation. What we didn't appreciate at the time is using a commensal control, we found that this organism produced almost the same level of inflammation as was seen with the pathogen itself. Now, with this in mind, over the last several years, we've spent a lot of time developing model systems to look at how the immature human intestine interacts with colonizing bacteria. As on sabbatical at the Pasteur with Danielle Levard, we developed a primary cell line, which we've used for the last decade, and in conjunction with Andre Caroni, developed the conditionally immortalized cell line. We've also shown ways to use organ culture from resected intestine and fetal intestine to measure perturbation, and we've established an ex vivo model for how the intestine functions in utero by transplanting portions of fetal intestine into the subcutaneous capsule of an immune-deficient mouse and then allowing it to reepithelialize, and we can then look in an ex vivo fashion at various perturbations. What we showed a number of years ago using our cell lines, using our organ cultures, was that when an exogenous stimulus or an endogenous stimulus was used and inflammation was measured by measuring mRNA and secretion of IL-8, we found that the fetal intestine was considerably greater in the inflammatory response. This is kind of a general phenomenon. The more immature intestine tends to overreact to stimulus with excessive inflammation. Now, subsequently, we were trying to define the mechanism of this process. So we looked at the fetal enterocyte genes for the innate inflammatory response. And what we showed was that there was an increased expression on the surface of toll receptors, an increase in the expression of their signaling molecules and transcription factors, and at the same time an underexpression of negative regulators. Here's what we showed specifically using the xenograft model. As you can see, TLR2 and 4 are overexpressed in the immature compared to the mature, and the negative regulators IRAC-M and TOLIP are underexpressed. We felt this was probably a partial explanation for the excessive inflammatory response. That is, that the fetal enterocyte has an immature innate immune response. All right, let me suggest that we might have a way of dealing with inadequate colonization by using probiotics as a surrogate. I suggest that by the addition of probiotics, we can move the imbalance into a restored balance system and hopefully can prevent disease. And let me illustrate this again with a couple of examples. A large clinical trial was conducted in Finland looking at allergy-prone mothers given LGG in the latter stages of pregnancy and then during lactation to see if there was an influence on the expression of allergy. Now, this particular study, unlike the one that we all have quoted, which was a smaller study, didn't show a huge difference in mothers who were allergic who delivered vaginally. What it did show, however, was a striking difference with the use of probiotics in mothers that were delivered by cesarean section. As we mentioned, necrotizing enterocolitis is the clinical corollary to the excessive inflammation that we see. Using our xenograph and stimuli, both endogenous and exogenous, as I've mentioned, we had a striking increase in inflammation. When we took two probiotics, Bifidobacteria infidus and Lactobacillus acidophilus, which had previously been used in a clinical trial in Taiwan to help prevent necrotizing enterocolitis. When we took these organisms, grew them, and then took their culture media and exposed them to the xenograph and then stimulated, we found that we could strikingly reduce the inflammatory response. when we went back and looked at the influence of these secretions on the genetic expression of the innate immune response, interestingly enough, we found that it caused a reduction in the expression of toll receptors and an increase in the expression of negative regulators. We think this might be a partial mechanism by which these probiotics are effective in helping to prevent this condition. All right, let me try to summarize and conclude what I've tried to say this morning. I've suggested to you that initial colonization is influenced by environmental factors, principally by the neonatal diet. The colonization is necessary for the development of intestinal host defense, that inadequate colonization can result in increased infections and immune-mediated disease, and that adequate colonization needs to occur in the immediate postpartum period. Finally, I've suggested, from a theoretical perspective, that inadequate colonization can be overcome by the use of probiotics as surrogate colonizers. Now, the work done in our laboratory has been done by a number of faculty that remain, some fellows who are previously there, and fellows who've gone on to develop their own laboratories, and then an ongoing collaboration with the cell biologist Andrew Caroni. Thank you very much.